Hello and welcome to Nikolai's genetics lessons, and today I got a problem from one of my subscribers, and here's a problem. If genotype homozygous for genes B and B is crossed to the genotype that is homozygous for both genes, and the F1 is test crossed, what percent of the test cross progeny will be homozygous recessive if the two genes are and for variants. So the first variant, unlinked. So how we are going to solve, and by the way, as usual, I recommend you to pause video here, try to solve this problem on your own first, and when you would be ready, you can run video again and you can compare your answer with my answer and explanation. So what happens here? So we have two genotypes. Parent 1 have genotype that is capital A, capital A, capital B, capital B, and we cross this genotype of parent 1 with parent 2 genotype, which is small a, small a, small b, small b. And I believe that 100% of you know that this is going to result in F1 generation that is going to be capital A small a and capital B small b. For the gene A, so gene A, first parent only can give dominant allele, and parent 2 only can give recessive allele. We know for sure that F1 generation is going to be heterozygous for the gene A. As for the gene B, parent 1 only can give dominant allele B, and parent 2 only can give recessive allele B. That's how we know that for the gene B, F1 generation is also going to be heterozygous. So we know genotype of the F1 generation. Now let's return to our problem. So if these two genotypes are crossed and F1, this is genotype of the F1 generation, is test crossed, so this is where students may get their first mistake. So many people do not understand what test crossed means. Test crossed means that we have to cross this genotype with known genotype and phenotype, and genotype have to be homozygous recessive for both genes. So that means that we have to cross with homozygous for the gene A and homozygous for the gene B. So again, this is going to be parent 1 in the second cross and parent B. And what we see here, we call this test cross, because we cross one genotype, sometimes it can be unknown genotype, with known homozygous genotype, homozygous recessive for both genes. It can be 3 genes, it can be 1 gene. So this is going to be second cross, and this is exactly we see here what is described in the first sentence. So what is the outcome of such a cross if genes are unlinked? Now let me show you different picture. This time let me draw chromosomes that we are going to get here. For example, as you know, parent 1 can produce following genotypes. For example, first variant would be capital A and capital B. Second variant can be capital A and small b. So capital A and small b. Next variant can be small a and capital B, small a, capital B, and the last variant of the gametes can be small a and small b. And parent 2 only can produce one type of the gametes. Parent 2 only can give recessive allele A and recessive allele B. Again, gametes are haploid. So parent 2 only can give alleles small a and small b. So now let's draw a Punnett square, and our results would be as follows. So now I want to show you how to solve this problem using Punnett square, but also I'm going to draw chromosomes and will show what's going on the chromosomal level. So we expect that genotypes of the next generation, this is going to be F2 generation, and genotype is going to be as follows: capital A, small a, capital B, small b, capital A, small a, and small b, small b, small a, capital B, small b, and small a, small b, small b. And the frequency is going to be 25%, or 1/4 if you like fractions, 25%, 25% as you see, and 25%. Again, what is our question? What expected frequency of the genotype that is going to be homozygous recessive for both genes? So as you see, 25% would be our answer. So question P, unlinked, give us 25% for, again, let me return to the question. What percent of the test cross progeny will be homozygous recessive for gene A and B? And as you see, 25% of the F2 generation would be homozygous recessive for both genes. So let's take a look at our answers. So this is question P, and we got an answer 25%. And we have answer 4, which give us P25, and answer 2, which give us P25. So we can cross right away. Answer, let me just put a cross here. Answer 1 and answer number 3. And now let's, instead of moving to the second question, let me show what actually happened here on the chromosomal level. The first question would take longer than the rest. To answer second question, third and fourth would take just a minute each, but let me spend a little bit more time on the first question so you would know better what's happening here. So on the chromosomal level, what is the genotype of the parent 1? And actually this is how chromosome would look like. So parent 1 would have one chromosome, and here we have allele A, and here we have allele B. So dominant allele A and dominant allele B. This is one chromosome, and here, or let me change color, so you would know that one chromosome came from mother side, another from the father side. So both chromosomes are homologous. So let's say this is chromosome number 1. So if this chromosome number 1, this is also chromosome number 1, but this one we got from, say, mother side and this from the father side. Or if it is plants, still the same rules applies. And here we have also dominant allele A and dominant allele B. So this is a genotype of the parent 1. Again, dominant allele A and dominant allele B and dominant allele A and dominant allele B. And this is parent 1. And we cross with another parent 2. Parent 2. And genotype of the parent 2 was also one chromosome. In the same locus, it has gene A, but different version of the gene A, which we call allele. And parent 2 has on the same, say, chromosome number 1, at the same locus, recessive allele A. And at the same locus, on the same chromosome as parent 1, parent 2 has recessive allele B. And this chromosome parent 2 got from, say, also mother side, and parent 2 also has another chromosome from the father side, and also has allele A recessive and recessive allele B here. So now you see both these genotypes. Also, let me underline with green color allele A and allele B on chromosome from the, say, mother side, and also with blue color allele A and B that parent 2 has on the other chromosome from the father side. Now, as you see, no matter which chromosome parent 1 would contribute to F1 progeny, so F1 progeny here, it can be any of these chromosomes. By the way, even if crossing over here happens, nothing actually happens. We still would have dominant A and dominant B, even if crossing over happens here. I show here with different colors, so you may think that, oh, it's going to be red dominant A and yellow dominant B, but actually it doesn't matter because the sequence is going to be the same. So it's still going to be dominant A and dominant B. Even if in parent 2 crossing over here happens, but as you see, these alleles A and B on both chromosomes are identical. And even if crossing over here happens, we still would have a chromosome with recessive allele A and B. So let me show it. Say by random chance, F1 progeny got this chromosome and dominant A and dominant allele B here, and from the parent 2 also by chance progeny got this chromosome with recessive allele A and recessive allele B. Again, both of them can be result of the crossing over, but still would have the same two variants of the alleles. Or if even crossing over didn't happen, it can be whether this chromosome or this chromosome, result would be the same. So this is not important. And this is genotype, as you see, of the F1 generation. So again, F1 generation is here, F1. Do you remember? Dominant allele A, recessive allele A, and dominant allele A, recessive allele A, and dominant allele B, and recessive allele B. So this is genotype of the F1 generation. And we cross it with another genotype which is homozygous for both genes, and we call this again test cross. So let me show this cross also here. Which color to choose? Okay, let it be yellow color here, and we have again two chromosomes. Number one, this is the same chromosomes, just different parent. So this is F1 generation, this is test cross. So we cross these two parents, but parent of the test cross is homozygous recessive for both genes. So would have small a here and small b here, small a here, and small b here. And this is what we call test cross. And this is exactly what I show here. Now you see it's on the chromosomal level. Now pay attention what happens here. This is two diploid parents. But these two diploid parents have to produce haploid gametes. So one of the parents have to be female, another parent have to be male. And it doesn't matter which one is which, whether this female and this male, or vice versa. And very interesting thing what's going to happen here. Again, this parent only can produce one variant of the gametes. Even if crossing over would happen anywhere here, once, twice, 3 times, it doesn't matter. This parent only can produce one type of the gametes, which is going to be chromosome with recessive allele A and recessive allele B. But this parent can produce much more variants. Take a look. Before this parent would make gametes, its genome, diploid genome of the germ-line cells would make copy of each chromosome. And this is also true with this parent. I'm also only showing what's going to happen here, because as I said, crossing over doesn't affect type of the gametes that this parent can produce. All the gametes would be of one type, but this parent can produce gametes of 4 types. So take a look what's going to happen. So this chromosome is going to make copy of itself, and this chromosome is also going to make copy of itself. So this is first stage of the producing gametes of the meiosis, and these two chromosomes we call sister chromosomes or sister chromatids, and these two we also call sister chromatids, and together this is going to be two homologous chromosomes, not identical. These two would be identical, these two would be identical, but these 4 would be homologous, so not identical. For example, again, on this chromosome we would see dominant allele A, and here at this locus we also would see dominant allele B, and another chromosome, because it is identical copy, would have small b allele here and small a allele here. So recessive alleles. So what type of gametes such, by the way, these 4 chromatids would form one chromosome, which we call tetrasome, which would consist of 4 chromatids, 2 sister chromatids and 2 homologous chromosomes. And we are going to get following variants. As you remember, one variant would be intact chromosome with dominant allele A and dominant allele B on it. Another one would be intact maternal or paternal chromosome with recessive allele A and recessive allele B, and also between these two non-sister chromosomes we can expect a crossing over. The greater distance between locus A and locus B, the more chances that crossing over happens here in many places, so more chances that crossing over would happen. And imagine if allele or gene B and A would be very close to each other, then we expect that no crossing over would happen at all. Or if we just make them closer to each other, then we expect crossing over would happen, but not as frequent as, for example, when this allele or gene A and B would be on the tips of the chromosome. So basically, when allele A and B would be at the tips of the chromosomes, we expect that they would behave as if they would be on the different chromosomes. So we expect that 50% of the crossing overs between these two non-sister chromatids would result in crossing over and new variants of the chromosomes. So the first variant would be with dominant allele A and with recessive allele B here. And another variant would be with recessive allele A here and also with dominant allele B here. So as you see, we got 4 variants of the chromosomes with different genotypes. And genotypes are as follows: capital A, capital B, capital A, small a, capital B, and small a, small b. And we expect that ratio is going to be 25%, 25%, 25%, and 25%. So exactly what we got here. Again, take a look. Parent 1 produce 4 variants of the gametes, and parent 2 only can produce 1 variant of the gametes. Again, now you see that the answer for the first question would be 25%. Now let's return to the second question. So, what if completely linked, so no crossing would happen at all? And now let's return again to this picture. So what if gene A and B would be completely linked? And let me just show here in the middle. Again, this chromosome, it doesn't matter. I just use this color, but doesn't matter here. And imagine we have gene A here and gene B just next to it. I even don't have space to show it, so I show it on the different side. So gene B would be here. And that means that no crossing over would happen here at all. So when we would have, for example, during meiosis, this chromosome would make another sister chromatid. Gene A and B would be so close to each other that even no crossing over would happen. So what is going to happen? And this is true for the second chromosome, homologous chromosome. It's just going to have different alleles. So let me use green color. It's just going to have different alleles. So recessive allele A here, recessive allele A here, and recessive allele B, and recessive allele be here. So as you see, crossing over may happen here, 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 here. If these genes A and B would be on the different edges of the chromosome, so we expect that crossing over would happen. But linked genes, completely linked genes, means that no crossing over between them happens and they would be inherited together. That means that such variant would produce only two types of the gametes. And the first variant would be gamete where we would have dominant allele A and next to it dominant allele B, and second variant of the gamete would be recessive allele A and recessive allele B. So basically we are going this genotype would produce only parental genotype chromosomes. So no crossing over would happen like in this example. And when we cross with, again, when we do test cross, so this genotype, when we cross with this genotype, which only can produce only one variant of the alleles, what's going to happen? So now you have already idea what's happening on the chromosome level. So let me also show this test cross. So again, if we assume that allele A and B are very close together, so what's going to happen? Parent 1 would produce gametes capital A, capital B, and small a, small b, and parent of second parent of the test cross only can produce recessive allele A and recessive allele B. When we build simple Punnett square, you see only two variants are possible. So capital A, small a, capital B, small b, and small a, small a, small b, small b. So 50% and 50%. Now let's return to our problem. So again, if you remember, we should look for the progeny of the F2 generation, which is going to be recessive for both genes, gene A and B. Let's take a look. Again, what percent of the test cross progeny will be small a, small a, small b, and we already gave an answer to the first question. It is 25%, and now we can say that for the second question it is 50%. And let's take a look. So answer 2 can be correct answer, and answer 4, as you remember. So answer 2 give us answer for the question P 25%, answer 4 also give us 25%, but variant 2 of the answer give us for question Q 50%, and variant number 4 give us 0%. So we also can say that answer number 4 is wrong. So we can cross this answer, and we can give right away an answer that answer 2 is the correct one. But let me show to the end what's going to happen here, because this is very interesting if you would know what's happening here, because now we are going to talk about map units apart to genes, and this is very important, but not going to take a time at all. So what happens here? Question R: what if between gene A and B we would have 10 map units apart? So let me return to these chromosomes and take a look. at this model where we have 4 types of the gametes that this genotype produce. So this genotype produce 4 variants of the gametes, and every variant would be represented as 1/4 or 25%. And actually this is true only if these genes are unlinked, so basically on the tips of the chromosomes. And there is very good chance, 50%, that crossing over would happen here. We cannot have chances over 50%, so 50% is maximum, because if we would have more than that, that means that it would have not 1 crossing over but 2 crossing over. For example, one dominant allele would go to the different chromosome and then would return back with a second crossing over. So 50% is a maximum. So that's why we have 25, 25 here. Together, this gives us 50%. So now imagine that gene A and B are not on the edges, but closer to each other, but not as close as in previous example, where no crossing over happens at all. But this time, let's say we move this gene somewhere here. So this is going to be a new position. And of course, not on just these 2 chromosomes, but we also move here and here, because this is homologous chromosomes. And if we move gene on 1 chromosome in 1 parent, that means that in all parents of the species, this is going to be a new position. So, and imagine that frequency of the crossing over now would be not 50% but 10%. So how our numbers are going to change here? So 10% crossing over means that new recombinants would be 5% of this type, 5% of this type, together 10%, and 45% of this type, and 45% of this type. So basically, what 45% here means that this is chromosomes where crossing over didn't happen. Why it didn't happen? Because 2 genes are very close, and chances are slim that crossing over would happen here. That's why, again, 10 map units means that we have 10% of the crossing over. So these 2 new recombinant alleles, recombinant chromosomes, would make 10%. So 5% each, with dominant allele A and recessive allele B, and recessive allele A and dominant allele B. And this 2 would be maternal and paternal original chromosomes, which would represented here as 45% each. So this is going to be an answer to the 3rd question. So let us return to variance, and as you see, this question is R. What would happen if gene A and B would be 10 map units apart? And as you see, in this case, we would see that, again, let me return. In this case, we would see that we have to return to this table. So parent 1 would produce this type of the gametes, but now, as you remember, in different frequencies. And this would result in different frequencies in F2 generation. So we would have, again, 5% here. This is going to be recombinant, 45% here. This is going to be recombinant, and 45% here, and 45% here. This is original chromosomes from each parent. So this original, and these two recombinants. So again, we're interested in this variant, and as you see, 45% is going to be our answer. And let's check here, and again we have our answer 45%. Now let's talk about S. We know that now we only have to choose this line, answer 2. If you have this question on your exam, basically right after you answered second question, you can choose this as answer. But today I just want to show you all the calculations so you would know the theory. So let's proceed how we got this 38% here. And the question S, as you see, give us 24 map units apart between gene A and B. So if we would have 24 map units, that means that frequency of the recombinant would be 24%. So 10% means 10% recombinance and 10% frequency. 10%. 24% means that we are going to get 24% recombinance, 12% each type of the recombinants. So together they make 24%. So 100 minus 24% give us 76% divided by 2. That gives us 38% of this type and this type, which is not recombinant. Again, you remember that this one would be genotype, which is small a, and small b. If we return to this table, again, we can say that this is going to be 12%, 12%. Together it's going to be 24%, and this one would be 38%, and this one going to be 38%. And again, this is genotype, frequency, which we are looking for. And again, we see this number here, 38%. I hope this video was one of the longest which I made during my maybe last month, but now you would understand this topic much better. In textbooks, it is very hard to explain what's happening, and it's much easier to explain in the video because you see all the dynamics. So let me add answers also here. So 45% and 38%. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. And see you in the next video. Goodbye.